CD 91 series, which we have planned. So it is a pleasure to welcome the first speaker, Dr. Anil Tarigopula, who is a senior consultant, Coimbatore Medical Diagnostic Laboratory, a unit of Coimbatore Kidney Care and Research Limited, Coimbatore, Tamil Nadu. His areas of work are histopathology and cytopathology, immunopathology, immunodisease, transplantation, immunology, and histocompatibility, HLA typing, and his main work focus in molecular diagnostics is highlighting on molecular pathology of solid tumors and hematological malignancies, including diagnosis, classification, prognostic evaluation, and predictive markers in therapy, bone marrow transplantation, and he has also the experience of setting up the entire molecular diagnostic laboratory right from the conception of establishing the required clinical diagnostic test. And he has also trained on quality management systems. He is also a NABL active assessor in the area of genomics and transplantation immunology, immunogenetics, and histocompatibility. He is also a member of various professional academic societies like Association of Molecular Pathology, Molecular Pathology Association of India, American Society of Histocompatibility and Immunogenetics, Association of Practicing Pathologists of India, and Indian Association of Pathologists and Microbiologists. He is also, as I said, he's an active NABL assessor. So I welcome Dr. Anil Tarigopula for his first talk on next generation sequencing, MPS in clinical diagnostics and overview. So welcome, Dr. Anil.
Thank you, sir. Thank you for the introduction. And I would like to thank CAHO for giving me this opportunity to present a little bit about an overview on what NGS is and why we should not be scared of using it. I'm just trying to share my screen. There is a reason why I put MPS as a short form for next generation sequencing. We'll unravel it as we go. Now, NGS is just a technology which has been in use for the last 10 years or so. And we know that a lot of things in oncology and germline testing has become more prevalently used on NGS platforms. But before NGS also, we were doing certain analysis for cytogenomics. In cytogenetics, I mean, just understand that cytogenetics, we were looking at a broad view of the entire genome in the form of a karyotype. And you can also look at whole chromosomes, or if you do FISH, still you're looking at small regions of these chromosomes, and you can identify. But again, these, even if you do FISH, the size of the region that you're looking at is going to be in megabases. So it was not a very good resolution to look at on cytogenetics, but you have a whole wide view of the entire genome. So it has its own role even today. So in particular molecular testing, we were doing targeted analysis of various genes and regions of interest of genes at a high resolution by using either sequencing, Sanger sequencing, or by real-time PCRs. And we have to remember that since we cannot have we do not have a technology which can read a single strand of DNA, we have to do something called a PCR, which is to amplify the regions of interest so that you can be able to identify these regions of interest and their variants on one or the other assay formats. So in molecular, we were doing something called a polymerase chain reaction, and it is, again, to do a PCR, you need to know your specific region of interest, and that region of interest may be something like, it may be a short range or a long range PCR, depending on what you're looking for. But broadly, you're looking for variants in a gene in regions of about 150, 200 base pairs, or if you are sequencing the entire exonic regions, you'll do up to about 1,000 base pairs, but resolution was limited. I mean, the extent of sequencing of the size of the DNA region of interest was limited, and you can only look for specific variants which are known unknowns in your sample. You cannot look for unknown unknowns, and it could also be quantitative. And after doing the PCR, you have to visualize the products that you have got, the amplicons, on one or the other methodologies. The common ones used to be the agarose gel electrophoresis, where you just by size and charge on the DNA, it separates out and you know what size you have amplified. and you have capillary electrophoresis at a higher resolution of that, at a 1 nucleotide resolution you have on sequencer. And you also used to do a quantitative PCR where you have an amplification which can be captured real time by fluorescence, and you can read the graphs and analyze them. Sanger sequencing was the predominant sequencing technology before the NGS technologies came up. NGS started somewhere in 2005 and started coming into the clinic in 2010. And by 2015, a lot of people heard about it and they were using in one or the other um, therapeutic, decision making for therapy. Sanger sequencing, the limitation was that you can only look for variants in one specific region, one single gene, one single region of interest, up to about 500 base pairs. You used to have a good read length. And the commonest and most useful application of Sanger sequencing when we were looking for a germline variant in terms of whether it was a homozygous or a heterozygous variant. Because if you see the plot, it is again visually identified and manually read. So if a variant is less than 20% in that, you will not be able to distinguish the signal from the noise. So it's very difficult to identify somatic cancer variants. In fact, in 2017, the AMP-ISLC CAP guidelines came out clearly saying that Sanger sequencing should be avoided for lung cancer because you will not be able to pick up anything below 20%. And we know the cutoff required for any assay to confidently be used in clinic is it should be able to pick up at least 5% of the variant against a wild type. Because in tumors, you have tumor cells against a background of wild type normal cells. So you should your sensitivity should be able to pick up at least 5% most of the assays. Now, this is just to show you, like Google Earth is like your karyotype where you see everything. It's also very important. It's not, and always we need to understand that NGS may not be able to identify. It is only looking at the sequences. It's not looking at whole chromosome losses, structural abnormalities, gains of chromosomes, duplication, some large amplifications. They're not easily identified on NGS. So we still need all these. They have different purposes. Karyotyping is like your Google Earth, and your fish is like identifying a region of a state, and your Sanger sequencing used to be identifying, for example, identifying a certain location, a spot on your region of interest. But what happened with NGS was you got the best of both worlds. It could integrate a high resolution into the entire thing. You could identify variants in multiple genes in multiple samples at the same time with a high resolution. So you are able to amplify, you are able to sequence parallelly multiple regions of interest of multiple genes, and you can also multiplex them by DNA barcoding with multiple samples of different patients, and you still have resolution available at one nucleotide. So that was the advantage of NGS. That's why it has become one of the mainstays. One of the mainstays. It's not all in all, but it's one of the mainstays for sequencing today. Now, let us see the characteristics of NGS, which has made it very important. It's a high resolution. So data is similar to Sanger sequencing, but you get a lot of data. Sanger sequencing, you're actually identifying the sequence of nucleotides in the region of interest. But here you are able to get a very high level, millions or billions of copies, depending on the kind of throughput that you have. And you can identify at nucleotide level. Broad view, like for example, in G-banded chromosome, you can do the whole karyotyping. Similarly, you can do whole genome sequencing. Whatever DNA is available, you can sequence the whole thing in a particular population of cells by extracting the DNA subject into amplification and sequencing the whole thing. And you can also look at a targeted thing like in fish. So you can do a whole exome sequencing where all the coding regions of the genome are included in the NGS panel, and you can study all the coding regions of all the genes. Roughly, we have about 20,000 functional genes in humans. Of this, you can amplify, and it's like bird's eye view of the whole genome you can get. And you can also use panels where you can target tens of hundreds of genes. You have small panels for myeloproliferative neoplasms. You have larger panels which can identify predictive testing for colorectal and lung cancers. And you have much larger panels where you can even get a tumor mutation burden score and all that. So there are targeted sequencing. A whole targeted sequencing can be whole exome or whatever, and you have the advantage of a broad view and also high resolution. So what are the terms? Why I said it is massively parallel sequencing? NGS has gone through various generations. Now what we have first, second, third, fourth, and fifth generations of NGS available. So I think next generation, how long should we keep calling it as a next generation? So the functional and appropriate terminology most of the people now have been switching to is massively parallel sequencing. So you have parallel sequencing of multiple regions of interest, and you can do for multiple patients at the same time. So it is an appropriate name. So you can use all these interchangeably. I think instead of calling it next gen forever, we can probably shift to MPS. Now, NGS can be, I mean, the main types of disorders that we look for are whether a germline or a constitutional variant you're looking for, some germline disease you're looking for, or are you looking for a somatic disease? So these are the two types of testings. And always we should remember a germline or a constitutional testing is different from a somatic testing of cancer. So expertise required will be different. A routine pathologist will not probably do a germline or constitutional testing, which requires medical and clinical genetics to come into play, while somatic testing is much appropriate for a pathologist to do. So germline genetic testing, diagnostic for genetic conditions, which are inherited disease conditions, which are germline in nature. So you have mostly blood samples, buccal swab samples, all these going in for testing. You can either large genes. Advantage of this NGS is you can sequence large genes like BRCA1, BRCA2, CFTR of cystic fibrosis, and have one test doing the entire thing without missing out any of the variants. Second is you can do large or small gene panels, tens to hundreds of genes for inherited diseases, and you can study the exome or whole genome. Again, these are tests which can be done at a lower, you need a lower coverage. You need to have a lower amount of data for each base pair for you to make sense of it, because it's a germline. It will be either homozygous, heterozygous. So there is no need. Somatic is a little different in it's more complex in its application. Hereditary gene cancer panels. This is one application where germline testing in related to cancer is seen for predicting the risk of developing cancer. Now, somatic or acquired, these are basically these directly are interpreted as related to testing for cancer. And here you're looking for tumor specific variants or mutations which are acquired and present in tumor cells only. And most of this testing is standardized on formalin fixed paraffin embedded tissues, whether it is a cytology specimen or it is a tissue. It is always better to have a formalin fixed paraffin embedded tissue. And one thing to remember in cancer diagnostics is nothing should go directly to the extraction without going through the microscope, because it matters what you are testing. So always a pathology review is required for any block, any slide from which material is taken for processing diagnostic tests for cancer. So most of the use is the commonest use is to predict response to targeted therapies. Then sometimes you can also have a diagnostic information or a prognostic information, especially sarcomas. You can find fusions if you do an RNA-based sequencing. So always, you can always convert, you cannot sequence RNA, you cannot amplify RNA directly, but you can always reverse transcribe and produce your cDNA, complementary DNA, and subject it to the same DNA sequencing process and have fusions and gene expressions detected on your NGS panels. So panels can be of various sizes that we have seen. And why do you want to do NGS now? Because it is scalable. The requirement for testing, you cannot do individually. Now, for example, in lung cancer, you have at least 20 genes for which you have multiple variants with targeted therapies available, or they are available. I mean, those particular variants are you can recruit the patient into one or the other clinical trials across the globe. So for doing that, you need to have, if you do one by one gene, it's going to take forever. So you need to scale it up, scalable both in terms of the number of patients you do and the number of markers for each patient that you do by sequencing. And the rest of it is self-explanatory. And one thing is, in somatic cancer testing, I told you it's not the heterozygous or homozygous you're looking for. You're looking for a variant against a wild type. There is a lot of heterogeneity in the sample and also in the tumor itself. Like you have a tissue which comes to you, if it is a lymph node metastasis, you have about 100, 200 cells, which are tumor cells, and rest of it is lymphoid cells. So you're not likely to find the cancer associated variant or the somatic variant in the lymphoid tissue, which can contribute a lot of DNA to your sample. So that is one. Second is within the tumor, there is a clonal progression of there are subclones in the tumor which form over a period of time, which can confer resistance and become significant. So you need to have a sensitivity of 1 to 5% of mutant against a wild type. So that's how sensitive the NGS can be for your cancer variant detection. And the other thing is you have a lot of data which comes out of NGS, though only we may have about, let us say, 20 mutations known in the EGFR gene of lung cancer, which are classifiable. But you also detect a lot of novel variants because you have the entire sequence. You're actually sequencing. So you can either subject your data to further research, or if you have a novel variant, you can look at whether it is relevant or not. So these are some of the differences between how NGS is different from, I'm giving an overview. So first part of my talk will be about the NGS, second will be the operational aspects, and the third will be about the problems and challenges. So here, this is, I think most of the things we have already covered. So I won't go through the details of this particular slide, but we know by now that these are the differences. One more thing is in NGS, the analysis is through bioinformatics tools, which you should validate and use. So it is automated. And in case of Sanger sequencing, you get something called a capillary electrophotogram, which is visually, or it's a manual analysis. So you have to read the sequence and identify variants in that particular output. Let's go to an overview of what exactly is an MPS workflow or an NGS workflow. Like any, when you apply NGS in a clinical diagnostics, it is not very different from the way you do any other work in your clinical lab. It's more of a technology which is used to, it's like another tool in your armamentarium. Like you have IHC, you have other things. Along with that, you have NGS also. So specimen collection, the pre-analytical variants, all the variations, those things are very important. Once you have an appropriate sample available, you have to isolate the nucleic acid. This part is different from a routine laboratory, like very important laboratories like biochemistry or hematology, because almost always the primary sample is not going to be the analyte. So nucleic acid isolation, depending on the complexity of isolation, you may either classify as pre-examination process, or it can also be part of your examination process. So every sample, when you try to study the DNA or RNA, and believe me, if you're doing a clinical workflow, you'll have 24 to 50, anywhere between 20 to 25 kinds of extraction protocols in your lab, because you're going to extract from different types of samples, right from cell-free DNA to your say this blood to body fluids. So anything and everything you will have to consider when a representative tissue is available to you. So nucleic acid isolation is an upstream process of NGS, which is very, very important. The quality metrics are very important once you do your extraction. Next, you generate something called a library. I told you there are regions of interest. We're not, if you're doing a panel of, we don't want to read the entire genome. We don't want to shoot the whole thing and spend a lot of thing and get the data for 12 to 15 genes. So when you look at a region of interest, you have multiple regions of interest in your individual genes. A combination, a collection of all the regions of interest in a particular sample for a particular purpose is called a library. So basically, you have to enrich your areas. You don't sequence the whole. You cannot put directly the DNA extracted into the sequencer. So you enrich it in the regions of interest. So like I already told you, there is no technology to read one single strand of DNA or RNA. So we have to amplify that to the level of billions of copies, which can be done by PCR or by hybrid capture. You can enrich them and then amplify that. Following library generation, you have other parts of this workflow where you multiplex and other things. And finally, you put the pooled library into the sequencer. Sequencing, there are multiple modalities, multiple platforms. You can choose the platform based on the application and the throughput that you're planning. And this is part of the wet lab. Now, data analysis looks like the smallest string here, but believe me, it is 60% of your work, and 60% of your bench will go into analysis of the data that comes out. So the sample, the history of the clinical history, everything constitutes the information you get to do the test. The process is your actual process of extraction and sequencing, and then the data which comes out, the data cannot, it's in the form of reads, which has to be analyzed, and finally you have to give a report, which is again information to the clinician about what we found in the specimen. So it's like any other clinical laboratory. NGS is another part of your armamentarium in the clinical laboratory. So what is so from sample to library and NGS? No, one more thing is that NGS we don't we actually break down a long strands of DNA into smaller pieces, read the small pieces, and using the bioinformatics tools we stitch them back and align them to the parent the reference genome of the human genome for comparing and identifying the variants. So this library is basically generated is it has to be fragmented. It has to be in smaller base pairs, 150 to 500 base pairs. There are long read chemistries available, but most of the somatics you have to with FFP, formalin fixed. The formalin actually causes a lot of artifacts in the DNA when it is when it fixes it. We do not have anything better than formalin fixation for us to preserve the DNA and RNA for use in downstream as of today. And I'm pretty sure the things are well standardized, but the nature of formalin fixation causes a lot of fragmentation, a lot of cross links in the DNA, protein, DNA, RNA, DNA, all these are cross-linked. So when you extract, it gets fragmented anyway. So there are various ways in which you enrich, you fragment it further. Even the germline also we fragment because your chemistries are going to be mostly base. I mean, it's 200, 400 base pair chemistry, maximum 800 base pair chemistry for most of the instruments. So you have to break down your DNA into fragments. And before you pool the samples, individual samples are barcoded with specific index DNA sequences to identify individual patients. So all the gene regions of interest of a particular sample, they have one barcode at the end of their sequence that is going to be sequenced. And after adding these barcodes, you add something called adapters, because to sequence, you have to immobilize individual strands of DNA on your either glass plate or on ion spheres. So to immobilize those, there are oligonucleotide sequences coated on the ion spheres or on the glass slide. And there are adapters which are added at the ends of the sequencing, this library sequences, which will go and complementarily bind to that and immobilize the strand there for you to get a coordinate on the slide or on the ion sphere for collecting the data of that particular sequence when you run the sequencing instrument. So again, when you prepare a library, now you may have a misconception if I don't explain to you that again in this also individual libraries, the ion spheres which are going to be sequenced, or on the Illumina glass slides, the oligonucleotide, I mean, these regions of interest which are to be sequenced, they cannot be read as a single molecule. So before you actually go for a sequencing, there is something called library amplification, and you have a clonal amplification following that, where each of the DNA initially it is emulsified in such a way that one template DNA, the DNTPs and other reagents required to amplify this clonally on the bead. So at the end of the emulsion bead PCR, you'll have thousands to millions of copies of the same clone of DNA on each bead. So when it sequences, and the signal coming out from each sequencing flow will be strong enough to be picked up on your sequencer. So again, emulsion PCR or cluster amplification is required before you actually do the sequencing. Now, sequencing, these are dependent. I mean, the way you do it, the capacities and capabilities, the types of applications that you can have are all platform dependent. Means you have mainly 3 platforms being used in clinical workflows. Thermo Fisher has Ion Torrent, Illumina has its sequencers, and you have nanopore technology. And for tumors, mostly we use Thermo Fisher or Illumina. Nanopore can do some of the germ lines and also can do the HLA typing. And HLA typing, anything can be done on these other sequencers. Nanopore usually not used for somatic because you have long reads here. You cannot read fragmented sequences of DNA which you get from FFPE tissue. So this slide is very important. If all of you pay attention to this reaction which is going on here, this is the sequencing reaction. Call it whatever you want to call it, but this is one reaction. Everything you have a DNA template at the base, And on the template, while sequencing, you are going to create a complementary strand synthesized along the template of DNA that you have a region of interest. And when a nucleotide gets added, there are three, when you do a sequencing reactions, three things happen. One is the nucleotide, if it is complementary to the template's base, it gets bound to the extending the primer and extends that. So if you tag it with a fluorescence, you can pick up the signal of the particular nucleotide which has added to the primer. So you know the position that you are looking at, and that is how Illumina works. Then you have, whenever this DNTP is added to the extension of, contributes to the extension of the primer, a hydrogen ion is released. So if you look at the pH changes happening because of hydrogen ion release, that's what you measure in an ion torrent system, which is a semiconductor based sequencing platform. So basically the chemistry, the basis, the nature of the reaction is same across platforms. What you measure and how you measure is different. So technologies are different, but the chemistry is the same. So this is an example of an Illumina. Those who are interested can go and read more about the individual chemistries. We may not be in the scope of explaining the technologies here. I just want to give you an overview so that you are familiar with all the components of NGS or MPS. Now, like I told you, sequencing is one part of your work. Your wet lab is about 40% of your work. Depending on the complexity or the size of your panel or the nature of your study, the bioinformatics is going to be very, very important and can be complex. But if you break it down and understand and try to get the right tools for each of the three phases of analysis that you have, then this and the pipeline is validated and you have gone through the AMP guidelines for validation of each of the steps of your protocol for bioinformatics. I don't think you should, I mean, basically it is resource intensive, both in terms of consuming the time and also you have to get the right bioinformatics tools for this. So basically bioinformatics is nothing. It is basically getting the signals from the sequencer into a way that you can put in your report. So there are three stages to that. First is the primary analysis. Like I told you, in ion torrent system, you have a hydrogen ion causing a pH change, which is measured as an analog signal in the form of an electric signal that is converted to a digital signal, right? This conversion is, it happens in two ways. One is each a small chip of ion torrent system on which sequencing happens is coated. It has about, not coated. It has about millions to billions of wells. In each of the well, only one ion sphere with a clonal amplified DNA amplicon is seated, and that is a coordinate on the chip. So many coordinates, a billion or millions of coordinates are there. Each of the coordinate is an address for a particular amplicon that you are studying. So whatever happens on that on that bead throughout the sequencing run, where you do multiple flows, you can do 400 flows, you can do 500, 800 flows. Depending on the number of flows you do, you get the length of the strand. Depending on what you are studying, you will set your length of the chemistry, and you will have each coordinate contributing to one sequence. So it is recorded as something like .wells and .dat files, which are not readable by us. These are instrument proprietary files. Primary analysis, we do not visualize, we do not actually see. These are all happening on the instrument, on its embedded proprietary software. We can only do the outputs. The output of that, what comes out from the sequencer, is a FASTQ file. This is the primary analysis. So the sequencer, based on its technology, converts the signals it finds throughout the various flows and generates a sequence. And the sequence is presented now. What we earlier, there were different formats, but the universal format now is the FASTQ file. It's called the FASTQ file, and it has the sequence read. It contains the coordinate. It contains the details of the region that the amplicon address is known. It has 4 lines, okay, specifically 4 lines. You have certain descriptor for, it includes the sequencer on which it was run, the chip and the address of the coordinate on which the particular, this is the read. The second line is the read on which it has been, the read that was actually captured during the sequencing. And this is something which is optional. There may be some additional information. And the next line is the quality scores for each of the positions. So we know in our keyboard you have all these are ASCII codes, ASCII codes. So ASCII codes are used for associating the quality. Each position has an associate. This is a dash or a hyphen for C. So each position is associated, each nucleotide, each base in every single sequence has a quality code associated with it. So these are also called as Fred scores. So Fred scores, basically anything, it is interpreted like this. Fred score of 30 means the particular confidence of something being accurate or something being inaccurate is 1 in 1000. So a Fred score of 20 is there is a chance of 1 in 100 times that that particular nucleotide call may be inaccurate. So for all our analysis, we prefer to have a lot of data beyond Fred score Q20, and preferably in cancer, when you are looking for specific variants, the system should have a good read, and you prefer to have something more than Q30. Means the probability of something wrong, something being read wrong is going to be less than 0.001%, right? Then now you got the FASTQ file. The FASTQ file is the output. So input is your sequencer's raw data converted to a FASTQ file. So FASTQ file is a large file, but again, remember, we have multiplexed. So we have in the sequencer multiple genes, multiple patients going together for sequencing. So the addresses are all, each coordinate has a random address to it. Now, we also know that we have attached the barcodes, patient specific barcodes or the sample specific barcodes are tagged. So FASTQ file, you have demultiplexed files, means one FASTQ file per sample. So all the same DNA barcodes are clubbed together and released as a FASTQ file. FASTQ file is not a complete run file. It is a demultiplexed file. Each FASTQ file represents one sample with the specific DNA barcode identifier that you index that you have processed while preparing your pooling your libraries. So further to that, secondary analysis. This is a primary analysis. You got the exact sequences with the address and you got your demultiplexing done. So sample wise, you have got your FASTQ files, which contains just the data of the sequences you have read. It does not contain anything about a particular gene or which species it has come from. It's just a sequence read. In secondary analysis, there is something called mapping and aligning. So you choose the reference genome against which you will map it and align. So in a region of interest, let us say there is a 500 base pair region of interest, you're going to cover the region of interest with multiple overlapping primer pairs in such a way that you have amplicons representing different regions of that 500 base pair. So you have to align all these. They may not be aligned. So you have to align them to the region of interest, and then the whole thing is mapped against a reference genome, and whatever is a variant, irrespective of whether it is a synonymous, miss, sense, stop, whatever it is, you get something called variant calling format file. VCF file is the output of the secondary analysis. So you have FASTQ, which is the primary output that goes in for mapping and alignment against a reference genome, and that file is called a BAM file. These two files cannot be read. You have to visualize. Reading these files and trying to make sense of it is not possible. While the output of the secondary analysis is your VCF file. This contains information in 8 to 9 columns, which are readable. It will tell you which chromosome, which particular gene, what is the allele frequency, what is the normal reference genome nucleotide or nuclear sequence in that region of interest. What is the variant there? What is actually changed there? All those details will be there in the VCF. VCF file, you can visually read it. You can read and understand which gene, which region of the chromosome, which chromosome, all details will be there. So till BAM files, all these are bioinformatics tools, and VCF is also generated through a bioinformatics tools. There are a lot of filters that you have to apply for quality control. I'm not going into the details of that. That will be a topic for a separate session. And the variants on all the variants are clubbed together, and from there, by filtering the variants to identify what are significant for analysis, you generate something called a filtered variant calling format file. The variants of that file Now you need to make sense in a clinical. You cannot report this file as a result. So when you take these variants you have identified and look at the databases and try to identify what is the interpretative thing about it, what are the clinical features described for that variant, which disease is associated, going to the databases and checking whether there are approved therapies for that, all that is part of your tertiary analysis where you filter, prioritize, and interpret the variants. So it's very important. This part of it has to be done by a person who is aware of the disease and who knows the variants and who is updated about the clinical guidelines and the availability of various um, databases. The people should be familiar with which database you should. These are not very difficult databases to search. Anybody who knows Cosmic database will know that it's not very difficult. You have to get familiar to it, read the information there. The content of that database is only information for your disease interpretation. So final clinical report should be reported with interpretation. I'll come to the specifics of that later. Now, tertiary analysis you have seen. Now, I have not told you about the kind of variants you can detect in a single NGS run. You have something called HGVS nomenclature, which you should go through to identify what are the various sequence variants, what are the various variants you can identify on different technologies. It's a unified nomenclature system. So we know there can be a single nucleotide variants, there can be complex variants where insertions and deletions are present as indels, and you can have copy number variations, you can be multiple repeats of a particular sequence or a gene. So all these have to have different filters. You cannot analyze everything using the same bioinformatics tools because these are different types of variants. Single nucleotide variants, most of the times you get good confidence in reporting. Indels are also okay. Copy number variants are very challenging to report unless you have validated your NGS pipeline, bioinformatics pipeline to identify this with an orthogonal validation. So you filter and one more thing is there are a lot of variants that you'll discover in your VCF. Not all of them are relevant. So you have to filter out what are not important, and then you have to classify, you have to identify what is normal, benign variations within the population. So you have to go to the databases, that filtering happens. And there is something called tiering, which happens in cancer reports. There is tier 1, tier 2, tier 3, and tier 4. This is nothing but the AMP CAP ASCO guidelines, which recommend you to classify your variant according to the level of evidence available to classify it, and also whether approved therapies are available or not. So you can go and look at the AMP's guidelines for the tier 1, tier 2, tier 3, and tier 4 pattern of reporting for cancers. Similar in ACMG, you have likely pathogenic, benign, likely benign, and variant of unknown significance. There is a different classification for germline diseases. Now, tertiary analysis involves a lot of times what happens is we tend to send the samples out and we get a report which is generated by AI or just general comments written in plenty. But if you read a paper by Cree et al. In 2014, when molecular testing had come into practice, it was clearly mentioned that the report should have interpretation specific for the disease and the disease state of the patient for in which the sample has come to you. So a little bit of review of the clinical features, whether it is a primary disease, secondary disease, or it is a progress disease, what you are evaluating, all that you should look for, and a report with appropriate comments for the disease state of the patient. Now, I'm not going to go into details. Most of it we have seen. Then an overview, we have a wet lab, which is basically the bench work that we do right from selecting the sample, enriching the sample, extracting the DNA, submitting it for library amplification or library enrichment, then your library pooling of the libraries with indexing and barcoding, followed by sequencing. The sequencing, you get primary analysis, tertiary analysis, secondary analysis, and tertiary analysis. Finally, you have a report. This is the overall workflow in an NGS lab. By nature of work and by nature of the steps involved, the quality control requirements, this is classified by CAP as a high complexity testing. So adequate manpower with appropriate training and with oversight by the clinical practitioner like a pathologist is required for interpretation of data in an accurate way. Now, In general, we have, I mean, this basically, I will leave it. Technically and both operationally, you have certain advantages of doing. You can find all types of variants in one single sample. This is truly a multiplex. What is the definition of multiplex? It's one sample, one run, and multiple results. And I think this is an example of a high level of multiplexing that you have. So these are advantages both in clinical terms as well as in terms of technical. Technically, if you have to identify 30, 40 genes variants, each individual variant, you have to do that much sample. You won't have. For NGS, a proper ion torrent based thing, you need only 10 nanograms of input RNA or DNA from a representative area of the tumor. So a good quality DNA will give you a good report. So there are certain terms that you can familiarize in this talk. One is enrichment, method to enrich your DNA or library for genes or regions of interest. You're not going to study all the genes in all regions in all the things. So you have to enrich, there is an enrichment process. You can have amplicon based, you amplify the regions of interest using multiplex PCR. And mind you, these PCRs are not in terms of 4 or 5 targets. One single tube, you'll be amplifying with something like 800 primers, up to 800 primers, 200 to 400 to 800 primers. So it's a rainstorm or huge. I mean, it's a highly multiplexed PCR. So it's almost impossible for any laboratory in clinical field to design your assays and validate them. So the expertise required in that has requirements for good manufacturing processes. So it's always good to get your things from a reliable source and be able to verify your assay runs. Now, the other technique for enrichment is hybrid capture. And you can have other exome sequences. These are other terminologies that we use. I've told you about index barcodes where you tag all the amplicons of regions of interest of a particular patient before you actually pull them together. And adapters are something which help them to localize onto addresses on a solid phase for sequencing. Now, I told you about Q30 Fred score. Fred score can be a thing below Q20 should not be acceptable for any kind of cancer decisions. So quality score, I told you a little about it. The reads are nothing but sequence of each bead, each address on the you get millions or billions of reads depending on the throughput of your run, the chip you have used, size of the thing, kind of sequencing reaction you're doing. Each single amplicon is a read. While in Sanger sequencing, you had a bunch of DNAs of the same size presenting with a fluorescent signal. Here, every single amplicon, which is monoclonal amplified on the bead or on the glass slide in Illumina, you have one read for it. Every single, you get plenty of reads, millions and billions of reads. So it's a huge file that you get. Then depth of coverage, it's a number of reads for a given region of interest. So depending on what, if it's a germline, you're looking for either 0, 50 or 100% for a variant. So you're looking for maybe a 30x, 30 times if you read, you get a good confidence whether it is homozygous, heterozygous. But for tumors, cancers, I would not go for any variant reporting below 250 anyway, but preferable is to keep all your coverage above 250 for any variant you're reporting. 250 is the lowest, that is lowest of the lowest. If you do not have any other sample you want to, but ideally you should cross check it with another methodology. But 500 to 1000 is not difficult to achieve, and preferably the coverage is one single important thing. It is mandatory to report coverage for a particular variant in your report itself. So this is one important thing, because at lower coverage, the formalin induced artifacts, there is a C to T and G to A artifacts which can be seen with formalin fixation. Any sample which has less than 250 base pairs, you're likely to get a lot of artifacts filtering as actual variants. So you have to be very careful. So coverage is very, very important. Any report which comes to you, it should mention the coverage of that particular variant, that sequence which has been reported. Then FASTQ file, BAM file, and VCF file we have discussed about. Now, one example of how NGS has made a change. From 2012-13, we were all doing real-time PCR for 29 or 30 different common variants seen in EGFR for lung cancer. And it was a qPCR or a PCR, ARMS PCR, whatever you do, it is going to only identify the variants for which the primers have been designed, and the kind of multiplexing requires you to do the test in multiple tubes. So this particular patient, he had 2 foci of adenocarcinoma, and both tumors submitted for later on. I mean, this is at the third. In 2017, he had a test by real-time PCR, and this 2017 there was a progression in 2020, and you had another second tumor also in 2020. It had 2 tumors. Now, when they actually sequenced with NGS, they found variants L833V and this is the protein sequence variation H835L. These two are in exon 21 of EGFR gene. Now, these are actionable, and these were not detected earlier because we were not looking for those. They were not the region, they were not the mutations of interest, because you cannot have a qPCR, which it will be very expensive and it will require a lot of DNA input. And most of the lung cancer patients, all you get it is maybe one, or if you're lucky, you'll have 2 cores of tissue, and that too, some of the histopathology labs may just trim it off and you may not have much to extract from. So in such a scenario, it's very difficult to do so many targets and identify in so many tubes in qPCR. But using the NGS, it was possible to identify a compound mutation containing 2 variants in cis configuration in the same exon of the gene EGFR, which was actionable because these are amenable to gefitinib and afatinib targeted therapies. So this is in practice, we actually, when you start doing NGS, you'll be surprised. If you have been practicing molecular pathology from 2010 onwards, you will see initially we were doing simple amplifications and capillary electrophoresis followed by real-time PCRs. Now we are doing NGS. So we know that we detect better now, and we are able to offer better decisions to the patients. Now, limitations are also there. I have already told some of the things. They are expensive, involve setup for new labs. Small batch sizes may not be cost effective. So these are all run costing. This is not sample wise costing. It is cost sequencing consumer. When you have to pool so many things, it is the denominator for costing will be the number of patients you are batching up together. And coming to the limitations, technically also you have platform dependent, assay dependent. So it's not like one assay will do everything for you. You have to select your assays. You have to select your instrument also carefully for the purpose that you're looking for. Now, I'll quickly run through why I should think of getting NGS for my lab. Now, operationally, I'm going to talk operationally now. NGS has 2 major processes. You have wet bench and a dry bench. So you should address the capabilities that are required for specimen handling, NGS library prep, and sequence generation. Wet lab is not a very big concern for you because a lot of trained people are there. A dry lab, a dry bench requires a very collaborative effort from the technical staff, your bioinformaticians, and the pathologist. It's not it cannot be all 3 phases have to be integrated. You should have robust protocols to do the analysis. You should have good pipeline, which is validated, verified in your laboratory with the hardware and software that you use. So these are the requirements, and these are self-explanatory. Anything that we ask for, it involves cost. So stakeholders, there are a lot of things that you need to consider. And mainly, if you have a good hospital where your clinical colleagues, good means every hospital, every colleague is very important and dear to us. But there are larger hospitals where there can be a demand from clinical colleagues to have things done in a better way with a turnaround time which is shorter. So these are the things that can help you in deciding why to do it in your center. Now, there are going to be barriers and challenges when we face a lot of it when we are in an institutional setup. There can be, barriers need not be just resources, but one thing to remember is it's not just the instrument that is going to play a role. Instrument is only one part of your requirement. So when you plan, see, one very beautiful article I read in Archives of Pathology a few years back in maybe 2015 or so, from behind a paraffin curtain, where it was very by I think it was by Ed Uthman or somebody, and I think he was in Salt Lake City, Utah, Arup Lab. So he has written an article where he said the third priority for a pathologist, first being the patient care, second being the technology implementation, third being financially savvy. So a pathology department or a laboratory section which is going to implement this should be aware of how to draw a business plan. There is a toolkit available with AMP. You can all go through it if you are interested in setting up. Go through it, understand how you can prioritize your requirements and present it to the management for you to go have a smooth sail for getting your things done. Now, okay, because we have NGS now compared to 10-15 years back, we have an increasing knowledge of certain predictive markers, prognostic markers, and diagnostic markers. We have a standardized clinical practice as in ASCO, CAP, AMP. All of them carry NCCN guidelines. All of them have standard practices prescribed with integration of genetics and genomics information. Now, there are some other things. I won't go into details, but you should know what types of, when you set up an assay, not only setting up the lab, once you set up the lab, you have to identify the assays. So you need to know what you want to identify in your sample. So if it is a germline, you should know that you have to get your information of relevant genes of interest and targets of interest from germline databases, which you should be familiar with understanding and reading them. If it's a let us say it's a copy number variance. I told you it is a difficult thing. So you should get the copy number variant databases to give you guidance on identifying those targets. If you are doing cancer work, you should know the somatic variant databases like Cosmic, OncoKB, Civic, Intogen. These are all, and for hematology, you have some disease specific databases are also there. So we should be familiar with that. If you're doing guiding pharmacogenomics, there are resources that you should be familiar with. Then pre-analytical conditions. One very important thing when you run any department or anything in the laboratory is the sample. So pre-analytical sample types, you have whether you should know whether you're looking for what kind. The sample will decide. Also, the type of testing you do will determine what kind of sample you require, whether it is germline, somatic testing. I'm not going into the details for brevity of time. Now, then pre-analytical, your extraction plays a role. You should know how to do the quality control. Different types of applications require either a nano drop is not a universal thing. So, I mean, universal means it cannot be applied for all your applications. You have cell-free DNA also, for which you need something called a tape station. And for degraded, a little bit archived FFPE samples, you should do something called a DIM score using a tape station for you to determine whether it is workable DNA or not. Then you actually do the sequencing, the type of sequencer, the kind of run that you are going to do, the kind of chemistry on the sequencer you are going to use. You should be familiar with that. Then you have another thing is your turnaround time, the number of samples you do and the kind of coverage you do are all interrelated. If you go for a very high coverage, you can do a lesser number of samples. If you do not have enough samples, you will have a denominator which is very small and resulting in unviable testing. So all these things you need to consider. And most important for a laboratory person is going to be the quality control considerations. You should be familiar with what quality metrics to use at which level of testing. One is the sample level quality metrics are there. Then you have coverage and other things in the data analysis. Then what if it fails? If a certain region fails, if you have some of the regions in the regions of interest on a particular assay and a particular instrument may not be very easy to do. A GC rich region may be very difficult region to sequence. So if it fails, what you're going to do? You have to think of all these things. So basically what we are doing now in even our setting up the quality policy is, quality standard in our laboratory is risk evaluation. So you have to think about all these and you should be familiar with all these, spend time on this, learn from peers, and you can set up. It is nothing to be scared of, just that you need to know the specifics of all these and set up. And for pipeline considerations, primary, secondary, tertiary analysis, variant calling, and the interpretation, you should identify the right databases, right kind of reference tools for you to do the reporting. And you have something called nomenclature system that we should stick with, because nomenclature plays a very important role. Historically, some of the variants that were reported earlier, we are not able to understand which exact variant is it as per the new system, because we were not following a uniform thing. Now, there are a lot of things, a lot of questions will be there. This is how we all grow in the field. It's not something which we learn on a day, but we have to start it. I think the biggest challenge to implementing NGS in India is we don't have it integrated in our teaching schedules in our MD pathology and other MD biochemistry programs, MD microbiology programs. Each of them are a domain of knowledge, domain of application. So pathologists should know everything about a pathology sample and use it as a tool for diagnosis and care. Microbiologists likewise should use NGS in diagnosis wherever it is applied or wherever you are allowed to do it as per the standard guidelines. Same thing with the pharmacology department. They have to do a pharmacogenomic study or something. They need to be aware of that. And the clinicians, of course, they need not have the entire knowledge of how to run a lab, how to do work in a lab, but they should have another challenge is having clinicians to develop working knowledge of a laboratory, what to order, how to ask for clarifications, how to discuss with the laboratory. Those are also important parts that makes life easy. But that once it is integrated into routine curriculum, it will be easier. But there's nothing that stops anybody from learning. So I have learned after finishing my MD only. So I think we don't need to go to Timbuktu to learn, but we have to keep on updating our knowledge and seek for the right mentors for this. So I'm just running through the slides. I don't want to take too much time. There are a lot of interpretation reporting. And one thing is, after setting everything up also, because a lot of instrumentation, a lot of informatics tools are involved, just be ready to have something annoying you. It can be a random error in your lab. There can be issues with your samples. You might have received a sample which has been over fixed. There is too much of formalin cross-linking, too much of fragmentation of DNA, not able to amplify it. So these are all upstream processes which will really cause a lot of problems. Archived samples are very difficult to do. Original sample issues, then pre-processing issues, where you have a small amount of tumor in a large lymph node deposit. If you don't macro dissect, this is called microscopic macro dissection. So you look under the microscope, you map out the area on an HND, next 4 or 5 levels, cut 10 micron sections, scrape off the fragment of paraffin roll and put it in the DNA tube instead of taking the entire curl of paraffin into that thing, which will reduce the sensitivity of the assay. You should select the region of interest in terms of the presence of the representative. I mean, you should take only the representative tumor into your DNA extraction. Bad DNA, bad report. So that we should not forget. Then a pathologist must always be involved in estimating the tumor percentage and making sure that the block is representative of the disease that you are studying. Then sample related issues can be there with wrong samples, inadequate samples. Then one more major problem with cancer testing is decalcified specimens. If you use harsh acids, it degrades and destroys, it abolishes all molecular work. So if you use HNO3, HCl, you use formic acid for a prolonged period of time without fixation, those are going to, so there are ways in which you can do it using EDTA decalcification. Take a small chunk of tissue, representative tissue, decal it separately. Don't subject the entire tissue for this thing. Send the soft part component or soft component of soft tissue component of the tumor for molecular analysis. Various ways in which you can mitigate that. So it is very important to think analytically at every stage of your work. Now, tissue decalcification, how it affects. I mean, these are basically. So false negatives can be there, low level variant. I told you, you can, for us, you're talking about large amount of data which is coming for any particular region of interest. If you have fewer reads, the confidence of reporting in that is going to be less because it's a smaller sample. If you have a huge, if for all the regions of interest, you have a haystack and for one, you have a bunch of grass, you're not likely to represent what is actually there in the tumor. So you have to be careful when coverage is not adequate, quality of DNA is compromised. So these are all problems that you can have. And sometimes even nomenclature can play a role. So for example, here, I mean, these are examples of a wrong nomenclature. Earlier, previously used nomenclature. You don't know whether it is actionable or not. So always use, this is a HGVS nomenclature does not have a separate book. It's a website-based nomenclature system. It is updated live basis. And this is the website address, which everybody doing NGS or any other molecular work should be aware of. It gives very beautifully all the definitions of various terminologies used in nomenclature, defines very clearly, unambiguously, the variants, how you should report, how you should write the correct way of writing every at the coding DNA level, at the protein level, how to report, how to include a reference genome along with your report. coverage should be, all those things are mentioned in your nomenclature system. So we know nomenclature is important for all the laboratory people. This is one thing that we live with. So we classify disease, we have to have a nomenclature system. So it can play a havoc if you don't follow the right kind of nomenclature. And there can be issues with bioinformatics tools going bad. And there can be weaknesses in your assay. The assay that you have selected, sometimes you don't review what is the purpose of that assay, whether it is able to detect those regions of interest. It may say EGFR, doesn't mean the entire EGFR gene and all your regions of interest for a particular disease are covered. There is something called a bed file, which you should go and check whether whatever you want to study is represented in the assay that has been designed for you. So it's very important. Then you can have false positive artifacts and low quality of DNA, misaligning of homologous sequences. Examples are there, which I think anybody who takes up will get interested and look at it. And one thing very important, even for a pathologist who works in NGS, you have to know how to use certain quality metrics, certain bioinformatics tools. You should be able to visualize the data on an IGV like this and identify the region, whether it is a true variant or not. All those things you have to see. You cannot report a variant without visualizing on IGV. That should be the basis for your pathology practice in NGS. Okay, there are a lot of things that can go wrong, but this is not the day for that. So thank you for the opportunity.
Thank you very much, Dr. Anil, for giving a lot of insight about next generation sequencing, and especially the MPS, how NGS has developed into MPS and SBS. So thank you very much for your nice